चलिए, okay, so everyone, good morning once again. We are doing this chapter sexual reproduction in flowering plants. ठीक है? We have finished this plant kingdom and the last part of the plant kingdom was gymnosperms, which we did, and after that it was you all were supposed to study the angiosperms. So in spite of studying that cycle in a shorter way, we are learning in a longer way because we have to study the everything. That is why we are doing this chapter sexual reproduction in flowering plants, right? So we are on the next part of this topic. Today we have to do some number of important things, right? Last class में what we did, let's go for a brief of that. ठीक? Last class में we talked about microsporogenesis, that is the development of pollen grain. Last class में we also talked about macrosporogenesis, that is the development of embryo sac complete, है ना? Till the egg development. Yes. So very quickly I am going for that microsporogenesis and megasporogenesis again, and then we are going to talk about the pollination. Okay? So I will be asking the questions from everyone because you have done this thing. So keep on speaking. Okay, if we have to go for this microsporogenesis, this means the development of pollen grain and the male gametes, right? So यहाँ से, like you all have seen, this part was the filament and यहाँ पे this was the anther. सारा दोबारा रिवाइज कर लो so that if you have any confusion you can get through it. Between the anther, this is a bilobed anther, and this thing with green which I am showing, this is called, जल्दी जल्दी बोलो, connective tissue. Connective, right? Connective tissue word use करते हैं actually we don't use this word. This is just the connective which is joining these two lobes of the anther. These anthers, they are also having some lines which is making each lobe to look into two thecas, and that is why we overall called it a diathecous anther. Diathecous anther. Clear to everyone?
Yes, ma'am.
ठीक है? So just 1, 2, 3, 4. Total of these 4 bags we are having, है ना? And each bag को now we are calling, क्या बोलेंगे? Microsporangium. Microsporangium. Each bag को now we are calling microsporangium, and this is the region where sporogenous tissue is present. है ना? There are number of sporogenous tissues present. ठीक? तो sporogenous tissue is present. This means if I just take one of this, तो ये है number of cells they join to form a sporogenous tissue.
Yes, ma'am.
तो एक word I have given. What you are going to call to a sporogenous cell? sporogenous cell. ये फिगर नहीं क्लियर? यहाँ पर देखो अगेन, if you go for the section of that pollen का section करते हो, so it usually appear this butterfly shape, है ना? यहाँ पे again you are having that 4 bags, ना?
Yes, ma'am.
And this part is called the connective. यहाँ पे I am saying the number of these sporogenous cells are present here, which are making these sporogenous tissue. तो इसके लिए word क्या था? What was the word which I have used?
Tapetum.
नहीं, microcytes, if you remember.
Yes, ma'am.
Yes, ma'am.
Right? This is microcyte and sporogenous cell is the same. Acha, if you are watching about the walls of this, so outer wall, this is the epidermis, and then next to this, epidermis is the outermost covering which is protecting this overall anther. Then next to this are present 2 more layers. One is the endothelium, है ना? And between that is the middle layer. So 2 more layers are present, endothelium and the middle layer. Both of them are taking part in the dehiscence of the mature anther. Mature anther means an anther in which the pollen grains are totally matured. ठीक है? So endothelium and middle layer they participate in the rupture of the thecas. अगर जैसे ऐसे देखो, ये एंथर था ना?
Yes, ma'am.
यहाँ पे जब रप्चर होगा, so this whole pollen, this will come out through this ruptures. Got it?
Yes, ma'am.
Now one more thing, you are watching this thing, the sporogenous tissue. So this bags, these 4 bags, they are enclosed by a ball. They are enclosed by a ball, and this ball through which they are enclosed, this wall is not uninuclear. क्यों भाई? Usually 2 nuclei are present in this wall. Cells.
Nutrition provide करने के लिए.
Yes, they provide nutrition. What is this wall called?
Tapetum.
Tapetum. Guranshi, she has learned everything and she is speaking fast. And now she is a competition for you all. So keep on speaking. Be quick. So this is tapetum. Everyone please try to be on time in my class. Inside it, यहाँ पर भी tapetum है ना?
Yes, ma'am.
All these pores are joined by this tapetum. Inside this tapetum is present sporogenous tissue, which is made up of sporogenous cells. These cells, they have got a prominent nuclei. These cells, they have got a prominent nuclei. Single nuclei है, but a prominent nuclei is present. Right? अब क्या होगा कि one of this microcyte, ठीक है, this will undergo mitosis. When it will undergo mitosis, it will make number of microspore mother cells or pollen mother cells. Right? So number of PMCs are here. अभी तक this is the part of the sporophyte. This whole is the part of the main plant. Right? So what need to be the ploidy of this?
Undiploid.
Diploid. Very good. Means two sets of chromosomes, 2N. यहाँ पर भी they are having this 2N and PMC is also 2N. Pollen mother cell is also 2N. Then this pollen mother cell, correct Ankit, they are going for this meiosis. Meiosis आपको पता है, this is a two-step reaction. Meiosis 1 and meiosis 2. So as a result, post meiosis always 4 cells are produced, है ना? If cytokinesis takes place, then तो यहाँ पे cytokinesis is also taking place and 4 cells, है ना? They are produced and they are अभी joined with each other. The ploidy of these cells is N. Now they are haploid. Got it? Now they are haploid. And this whole 4 wala structure, this is called a pollen tetrad. This is called a pollen tetrad. यहाँ तक clear to everyone?
Yes, ma'am.
Just giving again a quick recap. पूरा pollen grain was there. ठीक है? Full of this stamen यहाँ पे है. A stamen, ठीक? Number of stamens they join to form androecium. ठीक? We are talking about a single stamen. यहाँ पे हुआ anther and filament. Middle of this is the connective. If you cut through this, you are going to watch this type of structure. And इसको भी if you go for transverse section, you are going to watch this type of structure. यहाँ पे you are watching कि 4 bags are here of the sporogenous tissue, right? Outer wall is the epidermis, then middle layer, then endothelium. Epidermis for protection, middle layer and the epithelium, they participate in the dehiscence of the anther. Then coming to this exact bag, which is also called pollen kit या pollen bag we call, ठीक? So यहाँ पे this is surrounded by the tapetum. Tapetum is usually binuclear, right? It is providing the nutrition to the sporogenous cells or microcytes. ठीक? Then यहाँ पे they have prominent nuclei. Then अभी तक this was 2N diploid part of the main plant. Then this will undergo mitosis to form many of its type. ठीक? So a lot of pollen mother cells are produced. Then they will do this meiosis to form a pollen tetrad. यहाँ पे no cell. Usually what happened after meiosis, the 3 of degenerate and 1 is remaining. ठीक है? But that thing usually take place in the female part. Since you know कि females usually they are kept less in number and male these gametes they are kept in more in number because they have to go and they have to fuse with the female gamete and there can be different modes of their movement to this female gamete. That is what we are going to talk about in the pollination. So that is why these microspores or the pollen grains they are required in abundant amount. They are required in more numbers. Got it everyone?
Yes.
That is why they are required in large numbers. So यहाँ पे अभी तक post meiosis usually as a result of meiosis 4 cells are produced है ना which are haploid in number. तो यहाँ पे भी a pollen tetrad is produced which is haploid in number. Like this है ना ये चीज है same चीज यहाँ बनाई है same चीज I have drawn here haploid. Now they are maturing. Now they are maturing. They are maturing. So if you see कि यहाँ पे they have a inner wall which is smooth in texture and outer to this they have a outer wall which is non-smooth in texture, which is rough in texture. Outer wall is rough in texture, usually acid. This outer wall, this is made up of most resistant natural material which is called sporopollenin. This is called sporopollenin. And and outer wall this is called enzyme, and the yellow से जो मैंने बनाई है this is called intine, this is smooth, this is thin, right? There is some gap somewhere here, there is some gap somewhere here, right? इतना भी ज़्यादा नहीं जितना बन गया है, ठीक है? There is some gap somewhere here, and through this gap a pollen tube comes out from the intine part. This gap is called germ hole. This gap is called germ hole. Before this, what happened exactly in this? कि अभी ये था एक just n था, right? Then this have divided into two. One large vegetative nucleus and the small generative nucleus. The generative nucleus is spindle shape, है ना? Is irregularly spindle shape. तो ये आपका generative nucleus. अच्छा, meiosis has taken place already. यहाँ तक meiosis is over. After this, whatever is happening, like यहाँ के stage पे n is dividing again into 2n ना? यहाँ पे n बन रहे हैं. Like 2 cells are here. This is also n. This is also n. So this is a result of mitosis. ठीक? Not the result of meiosis. So ये irregular shaped nucleus होगा and a larger one. This वाला cell is called vegetative cell. Vegetative cell. ये दोनों joined रहेंगे. These two are joined. तो generative वाला cell, this is like floating in the cytoplasm of this vegetative cell. However, this generative cell's own cytoplasm is thicker and denser. ठीक है? Clear है यहाँ तक? And now after this stage is seen. This stage is seen post pollination. After this stage is seen post pollination. Means when the anther is lying on the female part पे when it is lying, when the pollen grain is lying on this female part. So suppose it is landing here, एक pollen आ गई है, and now this pollen is trying to take out a pollen tube. ठीक है? This pollen is trying to take out a pollen tube. So what will happen in this? अच्छा, I have also said कि more yesterday कि 60% of the plants they shed their pollen grain at this 2 cell stage only, at this 2 nuclei stage only. But in many of them ये process होना है. In many of them they shed at 3 nuclei stage only. So either they are going to form the 3 nuclei before shedding or after landing. Anyhow they have to form this 3 nuclei. ठीक? अभी तक it is just 2 nuclei. Okay? Now just see these 2 nuclei, they will reorient themselves, and ये जो vegetative cell वाला है, this will come in the forefront of this pollen tube, and ये vegetative वाले cell के nuclei को now we are calling tube nuclei. The purpose of the tube nuclei is to guide the pollen tube to the egg cell, right? And ये जो आप देख रहे हो generative वाला, this is divided again into 2 nuclei by mitosis. Again by mitosis, and these 2 nuclei will be called male nuclei, या फिर they are the exact male gametes. ठीक? They are the exact male gametes. I hope till here it is clear to everyone.
Yes.
Anyone having any doubt? Ikra, Kavya, Gayatri? Anyone? Now, this mature pollen grain is going to land on the mature stigma. ठीक है? If the 2 of them are of the matching type, then only it will allow this pollen grain to take out its pollen tube, and this is what called pollen pistil interaction. This is what is called pollen pistil interaction. Because of this interaction, only the mature type is allowed to take out its pollen tube. If this is not of the matching type, so either the plant is not going to allow it to take out its pollen tube even, or if suppose ये not matching है and this is now taking out the pollen tube, so some barrier will come here and this pollen tube will stop till here only. एक तो it is not allowing it to take out, and second अगर it is taking out the pollen tube भी तो barrier will come and this will stop it till here only. Okay? And yes, suppose ये yellow वाले से I am showing ये matching है. This वाला is matching. Matching pollen means that is of the same species, and yes, the plants also want this pollination to take place. Then it will take out its pollen tube. It will take out its pollen tube. ठीक? तो यहाँ पे we have seen कि a proper embryo sac was present. This embryo sac was having the 3 cells on the chalazal end. That is, these 3 cells are antipodal cells. Antipodal cells, right? Middle में it was the polar nuclei which have now fused to form 2n or n plus n, and यहाँ पे 3 cells were present here. Now, say 2 synergids and 1 egg. 2 synergids and 1 egg. ठीक? This whole embryo sac, कुछ ऐसा होता है, like this is the part of the embryo sac which is now formed because of from this, and this overall part को अभी भी we are calling कि ये nucellus है. This is nucellus. This whole nucellus, this embryo sac, they are part of a ovule only. In a ovary, number of ovules can be present. We are just showing one. We are just showing one. So this is covered by some protective layers called integument. The ploidy of integument is 2n. Nucellus की 2n. Antipodals की n. Egg की n. Synergids की n. ठीक? यहाँ पर definitive nucleus की 2n. ठीक है? Okay. Now this is a point where this integument is not present. This point is called micropyle. Opposite to the micropylar end is the base of this embryo sac, and this area, this part is called the chalaza. This part is called the chalaza or the chalazal end. So beta, now I am going to tell you all one more thing. Concentrate here, a new thing. There are different ways of entries of this pollen tube. There are different ways of entries of this pollen tube into a mature embryo sac. Got it?
Yes.
Most common method. Acha, whatever method they are going to use, they are actually entering through the micropyle. There are 3 methods: porogamy, chalazogamy, and mesogamy. But whatever method they use, अब later stage पे they have to enter through the micropyle only. Entry is from that point only into the embryo sac. So suppose ये वाला pollen tube is coming, ठीक है, and pollen tube is coming, and this pollen tube, ये देखो ये ball है, this is the ball of the embryo sac, है ना?
Yes.
If this pollen tube is entering and like यहाँ से चला गया, in fact और better हो सकता है, this can be more better. ये देखो, wait, should I use one line only?
Yes, ma'am.
चल, यहाँ से start कर रहे हैं, and this is coming beyond this integument, ठीक है? And now it is entering through the micropyle. It does not touch the integuments. Integuments को I am highlighting with another color. दूसरे color से दिखा दे रही हूँ. Now I am showing this integument with this blue color. Okay? ठीक है?
Yes, ma'am.
And now just see the pollen tube, it has entered inside this embryo sac, है ना? It has entered inside this embryo sac via synergids, but the entry was through the micropyle only, and it has not touched this integuments. Integuments को cross करता हुआ आया है, right? And this is the most common method of the entry of the pollen tube. But at the end, just see they have entered through the micropyle only, right? This method of pollen entry, this is called porogamy. This method of pollen entry is called porogamy. ठीक है? Clear to everyone?
Yes, ma'am.
When it is not touching the integuments or any other portion, just directly side से cross करता हुआ it is coming and it is entering through the micropyle to fuse with this. This is called porogamy. ठीक? अच्छा. Anyhow, porogamy पे ही just check a thing more. So when it was bringing their nuclei, it was the tube nucleus which was coming first. ठीक? The tube nucleus was guiding the pollen tube to come till synergids. And synergids, they protect the egg cell, right? And they also have filiform apparatus to guide the egg to reach to the
Egg.
guide the main nuclei to reach to the egg. So the male nuclei is coming. This male nuclei, they are trailing behind. The male nuclei, they are trailing behind this tube nuclei. After this, के male nuclei आ गया है. This male nuclei, one will fuse with the egg nuclei to form 2N, and the other male nuclei will fuse with this definitive nucleus to form this PEN, primary endosperm nucleus. So this was a process since 3 nuclei
or syngamy.
very good. This was a process वहाँ पे जहाँ पे triple nuclei were fusing. This was called triple fusion. This is called triple fusion, and ये वाला is syngamy. This wale part is called syngamy. So together triple fusion and syngamy together, this is called
Double fertilization.
double fertilization. Very good. Clear to everyone?
Yes, ma'am.
This is called double fertilization. ठीक है? So entries की मेथड चेक करते हैं. वही सेम चीज है, राइट? यहाँ पे आई एम मेकिंग दिस. ये इंटीग्यूमेंट है, माइक्रोपाइल है, और इसके अंदर एम्ब्रियो सैक है. ठीक है? एंड ये आपका हो गया स्टिग्मा. Now suppose this pollen grain is landing, and a porogamy you have seen already, and this pollen grain is now coming crossing this antipodal chalazal end, right? इसको cross करती हुई आई, and then it is through this micropyle. ठीक? antipodal जो cross करती हुई आई, and then it is through this. तो chalazal को cross करती हुई आ रही है, ठीक है? So this वाला type is called chalazogamy. Chalazogamy. So most of the plants they were doing this porogamy. ज़्यादातर में was happening the porogamy. ठीक? But some plants they show chalazogamy as well. When they come through the chalaza, the pollen tube is coming through the chalaza. For example, anyone who knows the example? Casuarina. For example, Casuarina, this shows chalazogamy. ठीक, एक और आते हैं, come to the next method and the last method of this. ये इंट्यूग्यूमेंट्स, ये एम्ब्रियो सैक, है ना?
Yes, ma'am.
Suppose pollen grain is coming and this pollen grain is now coming, crossing this integument and entering through the micropyle. So if it is crossing this integument, this wala method is called mesogamy. Mesogamy. Some plants they also show mesogamy. For example, Cucurbitaceae family. जो आप cucumbers खाते हो और gourd etc. Cucurbitaceae family. They show mesogamy. ठीक है? Clear to everyone?
Yes, ma'am.
Anyone having any type of doubt?
Ma'am, can you repeat mesogamy?
Sure, beta. When the pollen grain comes and matches with the stigma, so they lays off this pollen tube. So if this pollen tube is coming without touching the integument and all, and then entering through the micropyle to fuse with the egg, right? So this wala was called porogamy. Okay. Then next wala, when they cross through this chalazal end, this wala was called chalazogamy. And when they cross the integuments, ठीक, अगर integuments को they are crossing, so this is called mesogamy. ठीक है?
Understood, ma'am.
Uncle done? चलिए, अभी तक we have seen different methods of this pollen tube entry. We have also seen pollen crystal interaction, ठीक, and the ovule. ठीक, ovule जो आपने देखा, this was like this. क्यों भाई? ovule ऐसा था ना?
Yes, ma'am.
This was like this. This is the most famous and the common category of the ovule. This is the most common and common category of ovule. ये क्या है? This is the
Follicle.
follicle. This part is the hilum, है ना?
Yes.
यहाँ पे integuments है, right? Like integuments, they have covered this. Integuments, they have covered this ovule, right? And this is the point. This is the micropyle, है ना?
Yes, ma'am.
And this part, this is the
Chalaza.
chalaza. ये याद है ना? This is the most common type of ovule. This is the most common type of ovule. Anyone who can tell me what is this type of ovule called? What is this? Very good. Get very good. This is the most common type of ovule, and this ovule is called an anatopous ovule. So most of the plants they have this type of ovule. ठीक है? जितना मैंने प्रॉमिस किया था कल बहुत सारी चीजें आज पढ़ा रही हूँ. ठीक है? This type of ovule, this is called an anatopous ovule. This is the most common type. This is the most common type. ठीक? तो यहाँ पे तुम देख सकते हो क्या है इसमें कि chalaza and the micropyle they are lying in one line, है ना? And ऐसा कुछ है like a type of 90 degree from the hilum. Follicle and hilum से they are little bit like this. And chalaza and micropyle they are lying in one straight line. Got it? But a second version is, चलो तुम लोग देख के बताओ. माइक्रोपाइल, चलाजा, फ्यूनिकल, ठीक है, और बीच में आ गया आपका एम्ब्रियो सैक। समझ रहे हो? तो सेकेंड वर्जन इज दिस in which the micropyle, the chalaza, the funicle, they are all in a straight line. They are all in a straight line. Very good, Ankit. This wala ovule is called orthotropous ovule. Orthotropous ovule. This is also called atropous. This is also called atropous. Okay, so this is the most common type of ovule. Okay, anyone who knows the example? Polygonum. Polygonum. Example में polygonum, beetle, they are the examples of this orthotropous या atropous ovule. Clear to everyone?
Yes, ma'am.
किसी को कोई डाउट तो नहीं?
No, ma'am.
Clear. Okay. Just check the movements, how it is like changing the shape. एक और category है, like ये again funicle and this is like ovule. So like one end पे micropyle, another end पे chalaza, and this is the funicle. So you are watching कि like it is placed in 90 degree, like इसके ऊपर funicle के ऊपर this is placed like this. कि micropyle and chalaza तो are in one line, but it is lying perpendicularly above to this funiculus. Anyone who can tell the name of this ovule? Amphitropous ovule. Amphitropous. Okay, this is called amphitropous. Example, lemna. Lemna क्या होता है? duckweed. Lemna, duckweed. ठीक है? Clear to everyone?
Yes, ma'am.
अब suppose this ovule is changing in shape just like a horseshoe. Horseshoe क्या होता है? ऐसा कुछ, यहाँ पे ऐसा कुछ होता है ना horseshoe.
Yes, ma'am.
हाँ? आप यूपिन भी बोलते हो मैग्नेटिज्म में.
Yes, ma'am.
horseshoe magnet होता है ना?
Yes.
Right. चलो, अब this ovule is like changing its shape. कुछ ऐसा कर गया है. Okay, यहाँ पे like chalaza, one side this is micropyle and this is funiculus. तो ये तीनों ही, three of them even, they are not in a straight line. अभी तक देखो straight lines आ रही थी, but यहाँ पर कहीं से भी straight line नहीं है, no straight line. Okay, this वाला, anyone who knows the name of this वाला? Sarcinotropous. क्या क्या पढ़ा? Anatropous, Orthotropous, Amphitropous, Sarcinotropous. Okay? Example, it is mostly seen in this Crucifery family. यहाँ पे आपको you are getting the example of mustard. उस family में this is usually seen. Mustard. ठीक है? Mostly seen in the Crucifery family, like a mustard. I hope the different types of ovules are clear to everyone.
Yes, ma'am.
यहाँ तक clear to everyone? ठीक है? चलिए, तो we have learned two things अभी तक के. We have learned the different ways of this entry of pollen tube, and we have also learned the different types of ovules, right? And We have also revised जितना भी कुछ था अभी तक के whatever we have done so far yesterday. ठीक है? ये सारी चीजें we have done. Now let's come to the task of pollination. ठीक? You all feel कि pollination is very easy. So yes, you all are true. This is a very easy process, but little bit lengthy है. So we are going to do this all in detail. चलो, let's start with the pollination. So yes, participation is highly required. Pollination. What is the meaning of pollination?
Transfer of pollen from anther to stigma of same or another flower.
Very good. बचपन से एक चीज we have learned कि transfer of pollen from the anther to the stigma. तो इसको लिखेंगे. Anther to stigma. अब बेटा, my बच्चा party, we have got two categories of plants, है ना? We have got two categories of flowers. So देखो, there are some plants like papaya, which is a unisexual plant. Complete plant is unisexual. If male plant is different, female plant is different. Or we have some plants who are having same, उसे हम कहते हैं bisexual plant है, right? के male flower, female flower, या a flower which is having both girls on it on the same plant. ऐसे भी plants हैं. So every category we are going to check. तो सबसे पहले what we are going to do with this pollination कि just divide it into two categories. ठीक? This pollination overall can be of two types. Self-pollination and cross-pollination. Very good. Self and cross. Technically, इनके दो नाम से, ठीक? Self-pollination means autogamy and cross-pollination means allogamy या फिर xenogamy. What is the meaning of gamos? What is the meaning of gamos? Word gamy मिल रहा है ना?
Fusion.
हाँ, exactly. In Latin it means marriage. It means marriage. तो एक ही घर की marriage या फिर दूसरी जगह जाके like after watching whole कुंडली and everything this marriage, है ना? Whether it is going to be suitable or not. The self-pollination means autogamy and cross-pollination means allogamy या फिर xenogamy. ठीक बात?
Yes, ma'am.
Clear? चलो. So let's first check this autogamy. एक flower है ना? We are taking a simple flower here. कुछ भी बना लो. And this flower is having, यहाँ पे उसका स्टिग्मा आपको दिख रहा है, ठीक है? Ovary style stigma, and also you are watching its stamens, है ना? Means you are watching its piston as well as the stamen. दोनों चीजें you are watching. So this is a flower. This flower you are going to call monoecious या dioecious. सबसे पहली चीज के all the floral whorls are here. First of all, this is a complete flower. सबसे पहली चीज I make you to clear this thing yesterday only, ना? कि what is a complete flower? So this is a complete flower. And this complete flower is having both of its whorls, both of its sexual whorls here, है ना? Means stamen is also here and pistil is also here. Okay? So when a flower is having both of its two whorls together. So only this type of flower can show technical true self-pollination. है ना? Only this type of flower can go for autogamy, but it is always not necessary that these flowers always move with autogamy. ठीक है? वो हम सीखेंगे बाद में, after the later stages. So only this type of complete flower, या a type of flower which is having both of its sexual whorls, that is a monoecious flower, can go for this autogamy. ये बात clear?
Yes.
When both of the reproductive whorls are here, that is the male and the female parts are here, then technically it can go for this autogamy. ठीक है? This is a male plant, this is a female plant, and both of them are having something. यहाँ पे जो flower है, this is of course a male flower, and this is of course a female flower. And when this type of pollination is taking place, this is called allogamy या फिर xenogamy और cross pollination. ठीक? Again I am showing a plant. ठीक? This is having one flower which is male and it is having another flower which is female. समझ रहे हो? Genetic constitution is same. एक ही plant है ना?
Yes, ma'am.
Genetics same, but technically one flower to other जा रहा है. When a pollination from anther of one flower to stigma of another flower, है ना? This is called allogamy, है ना? This is called allogamy. So what you see here, this is the true case. तो exact जो होना चाहिए principally, this is the allogamy. But this is also a allogamy, but genetically अगर हम देखें तो this is a autogamy, right? So we cannot clearly call it allogamy या autogamy. हालांकि this is also placed in the case of allogamy. So this is called geitonogamy. Spelling check कर लीजिएगा. This is called geitonogamy. This is called geitonogamy. Clear to everyone?
Yes, ma'am.
When the stigma is being fertilized by the anther of the flower, which is not the same flower but was present on the same plant. Plant यहाँ पे भी again bisexual है. Plant again still here is bisexual, but technically this is geitonogamy, a type of cross pollination. सबको clear?
Yes, ma'am.
There are some plants who prefer these things. Like किसी को कुछ अलग सी चीजें चाहिए होती है. Like Commelina, which is like called pansy, है ना? और कुछ examples हैं.
Oxalis, viola.
Oxalis, viola, Commelina. They go for two categories of the flowers. One is the jasmogamous flower. One is the jasmogamous and other is the clistogamous. So two type of flowers they have, jasmogamous and the clistogamous. Clistogamous, this flower guarantees only self-pollination. This flower guarantees only self-pollination. ठीक, यहाँ पर भी self-pollination हो सकता है, but this cannot rule out cross-pollination. ठीक, this cannot rule out cross-pollination क्योंकि this flower is a open version. But Clistogamous flowers, they are closed flowers. They are closed, like the anther, and the stigma is closed inside the petal. So no outsourcing of the anther can be done. So they only go for self-pollination. Chasmogamous is also a normal self-pollination वाला flower, but कोई भी फ्लावर अगर ओपन है तो दिस कैन नॉट रूल्ड आउट क्रॉस पॉलिनेशन. ओके, सो एग्जांपल्स याद रखिएगा. वोइला, कमैलिना, ऑक्सैलिस, दीज आर द एग्जांपल्स ऑफ दीज फ्लावर्स of plants which are having these two categories of flowers, that is the chasmogamous and the clistogamous flowers.
तो फिर मैम जो लोटस होता है वो भी क्लिस्टोगामस में आ सकता है, बिकॉज़ वो तो ऐसे लिखा है क्लोज.
लोटस खुलता भी तो है, ये होता है. ये तीन याद रखिएगा, दिस इज अ पार्ट ऑफ योर सिलेबस. ऐसे अदरवाइज दिस वी हैव अ ह्यूज बायोडाइवर्सिटी, है ना. So far it is clear to everyone. So far it is clear.
Yes ma'am.
Yes, ma'am.
चलो, come to the next thing. यहाँ तक we have seen. ठीक? इससे पहले we go for the agencies of the pollination and all. हाँ, बस जो normal plants of the self pollination आप देख रहे हो, normal open plant, अब broadly we can call them chasmogamous, and जो close वाले हैं उनको we can call clistogamous. ठीक, however they are highlighting it because it was present in one plant only. उसके अंदर ही two types of plants were shown. That is why they have highlighted it with two names. One calling chasmogamous and the another close वाला to be clistogamous. ठीक है, ये हो गया. So इससे पहले we can go for the agencies of pollination. ये वो वाले process में we start. Let's go for some other thing. हाँ, yes Shini, they are pollinated before opening. So let's go for something like it is called outbreeding. ठीक, outbreeding. Yeah, there is some plant like which is not compatible for self pollination. You know, okay, both self pollinations and cross pollinations, they have some aspects. Okay? Self pollination is a thing like which is carrying on continuous legacy. एक type है उसको they are continuing, they are not allowing the mixing, so that you can say कि one progeny can be maintained, one species can be maintained. But continuous self pollination can make in a loss of vigor of the plant. So that is why sometimes आपने देखा था like lower organisms में भी paramecium was going for cross pollination every 24 hours just to regain its lost vigor, है ना? So cross pollination is a preferred thing in a way कि it brings back the variation, it brings back the lost vigor, it is the source of lot of variations. ठीक? Different categories you are watching because of the cross pollination. But self pollination is not allowing these variations. So there are some plants in this outbreeding you will learn only this thing that there are some plants that they are having some techniques so as to go for cross pollination only. ठीक? So this is called outbreeding. So let's check out कि क्या क्या हो सकता है in this outbreeding. या फिर techniques to prevent self pollination. techniques to prevent self-pollination. ये चैप्टर अच्छे से तो सबको आ रहा है ना?
Yes, ma'am.
I don't think you are having any type of doubts. जो भी है that is getting clear.
Yes, ma'am.
चलो, so एक सबसे पहली चीज, सबसे first thing is this genetic incompatibility. Even एक same flower है, वहाँ से pollen can land on the stigma of the same flower, but the plant is like having a type of genetic incompatibility. It is not allowing the pollen tube to come out. या फिर अगर आ भी रही है, then it will disintegrate. या उससे, है ना? This is a way, like this is called genetic incompatibility. ये जो भी चीजें हैं, they are not genetically compatible to each other. तो geitonogamy भी sometimes it even don't allow. Just not the autogamy, but sometimes this genetic incompatibility doesn't allow geitonogamy only. It prefers, क्योंकि टेक्निकली तो geitonogamy भी अगर हम देखें तो जेनेटिकली तो वो भी चली जा रही है in the same ही कैटेगरी ना, कि जीन सेम है. So and they want variation. They want variation. That is why is genetic incompatibility में even more of the plants that don't allow geitonogamy only. They go for this cross pollination only. तो ये एक हुआ कि वो जेनेटिक कंपैटिबल नहीं है एक दूसरे के लिए. ठीक है? अच्छा, in order to go for self pollination अगर होना है, so also this thing can also be not ruled out कि the anther and the stigma they should mature at the same time only, है ना? And means कि anther should mature and stigma should become receptive at the same time only, है ना? तभी तो self pollination, even if they are in the same flower, but the anther and the stigma they should mature at same time. तभी तो the pollen will land on the stigma and the stigma which was receptive will receive it, है ना? So if they are not maturing on the same time, then also this self pollination be avoided, है ना? समझ में आ रहा है?
Yes, ma'am.
Anther and stigma not maturing on same time. अच्छा, if we are watching point number 2 where I am saying anther and stigma not maturing in same time, ठीक? But suppose let's point number 2 is ruled out. कि anther and stigma they are maturing even on the same time, but they are not genetic compatible to each other. So this is the second way, है ना? Got it everyone?
Yes, ma'am.
ये दो points clear?
Yes, ma'am.
Okay. Means I am talking about a flower which was having both of these two girls together, right? A plant which is bisexual, ठीक? Monoecious flower की बात कर रही हूँ, where both of these reproductive bulbs were present on the same flower. So these two things can be done to avoid this, to avoid self pollination. एक कि भाई ये mature ही ना हो same time पे, और दूसरा even if it is maturing, तो they are not genetic compatible to each other. ठीक है? There is some another ways also. There is some another ways also. ये आपको सीखना बहुत important है. NCERT is not giving the correct verdicts, terminologies. They are not given the correct one. उसको सीखिए. There are two methods. One is called dichnogamy. And another is heterostyly. Dichnogamy में two things. one is the protandry and another is the protogyne. प्रोटोगाइनी, ठीक। हेटेरोस्टाइली में डायस्टाइली एंड ट्राइस्टाइली। ये इसी का पार्ट है, लाइक एंथर एंड स्टिग्मा नॉट मैच्योरिंग ऑन द सेम टाइम। तो इसमें डाइकोनोगामी कैन टेक प्लेस, ठीक। इसमें डाइकोनोगामी कैन टेक प्लेस, लाइक वन इज द प्रोटैंडरी, इसमें एंथर मैच्योर्स फर्स्ट, है ना, एंड स्टिग्मा मैच्योर्स लेटर ऑन। सो बोथ ऑफ देम आर नॉट टुगेदर मैच्योर। प्रो मतलब फर्स्ट, एंड्री मींस एंड्रूशियम, तो एंड स्टेमेंस एंथर दे आर मैच्योरिंग फर्स्ट। और प्रोटोगाइनी मींस फर्स्ट gynoecium, gynoecium means pistil या ovary matures first। क्लियर हो गया ये सबको? This is clear?
Yes, ma'am.
Very good. Another thing is the heterostyly, ठीक? Another thing is the heterostyly। heterostyly में there are some cases, like अगर ऐसे देखो एक फ्लावर है, ये फ्लावर का बेस यहाँ पे डायस्टाइली है, ठीक। तो ये हो गया स्टिग्मा, ठीक, बट ये नीचे है, this is not able to fertilize it, right? एक और हो सकता है ट्राइस्टाइली में, like different methods होते हैं of this, कि कभी-कभी ये ऐसे है and then like some are here, ठीक, and some are here, and there is another way कि ये ही जाके लंबी हो जाए, this will become long, इन सब से ऊपर हो जाए। So different methods, like heterostyly is a method कि style and stigma they are not matching in the sizes with each other, so that this cannot fuse it easily, so as to avoid this orthogamy, avoid self-pollination. The dichogamy and heterostyly is clear to everyone?
Yes, ma'am.
ठीक है? So these are some of the methods, some of the techniques to avoid self-pollination. Anyone who is remembering any other method to avoid self-pollination? Yes, Shirin, write in the chat box. What do you exactly mean by this? Prepotency. Can you explain this? Like so far the NCERT is only describing about this much. So these are some of the natural methods. These are some of the natural methods how they are avoiding this. But like human being, these days you will learn this in future when you will come to the class 12 portion में कि continuous of this will lead to inbreeding depression. Yes. This continuous inbreeding will lead to a type of depression. वही जो मैंने आपको चीज बताई कि they are not so much active, ठीक? They are losing their potential. So that is called inbreeding depression. So उसी के लिए cross pollination is required. उसी के लिए cross pollination is required.
मैम इसमें आउटब्रीडिंग टेक्निक्स में बैगिंग भी तो हो सकती है।
हाँ, मैं आ रही हूँ वहाँ पे, ठीक है। So the scientists, what they do, they can go for artificial hybridization. ठीक, अभी बहुत सारी चीजें हैं अभी, लाइक भी आपको एक चीज मिली होगी, लाइक पोटैटो एंड टोमेटो टुगेदर दे आर हाइब्रिडाइज्ड ना, नॉट जस्ट द प्लांट्स, भाई देखो there are many plants like which are having naturally cross pollination by having the two sets of the plants, like male plant and female plant, जैसे पपाया है, ठीक है। But another case is the artificial hybridization. उसमें like if you go and fuse with this tomato and potato together, तो a different thing is found, है ना। Usually आपको एक मिलते थे ऐसे, when I was young I used to eat those watermelons which were very big and green, dark green in color. But gradually I become adult, I used to watch a watermelon which is having little bit lighter green texture and some light dark green stripes, and the seed size is also reduced, and earlier that greener wale were having the bigger seed size. So this is all like they are trying to match different species just to get new thing, है ना। or like potato and tomato का match is usually called tomato, potato and brinjal का match is called tomato. So different matchings भी they are, yes Reshma, different matchings भी they are doing in this artificial hybridization. So there are some techniques to do this artificial hybridization. And some of these techniques are used since the time of Mendel. Richard Johann Mendel also used these techniques. And he used these techniques to with his pea plant. So one is this called, very good, one is called emasculation, and another is bagging. मुझे पता है तुम सबको पता है, बट मेरे ढंग से समझने की कोशिश करो। emasculation, masculation is from the word masculine, मेल के लिए आ रहा है, दिस इज कमिंग फॉर द मेल। अब emasculation मींस रिमूवल ऑफ द मेल पोटेंशियल, है ना। सो दिस मींस मेल पार्ट वी हैव हियर इज द एंथर, तो ऐसे आपको याद रहेगा, दिस इज द रिमूवल ऑफ द एंथर, ठीक, रिमूवल ऑफ एंथर। तो सपोज there is a flower which is bisexual, ठीक है, and they want to do a artificial hybridization for it. So what and जो भी था उसमें anther and the stigma that was of a type which was maturing on the same type and that was genetic compatible to each other. इस सारी cases of self pollination that flower was fulfilling, ठीक। अब क्या करा गया कि like they have removed this anther, that is masculine portion को remove किया गया with the help of forceps, right? So this is called emasculation, and then जितनी भी उसकी anthers थी, all of that anther are now removed. It means कि अब they are avoiding it, self pollination is not allowed, but they want to hybridize it with the pollen with the anther of their choice. So any pollen from the wind या insect can also come and land on the flower, which cannot go for self pollination but can go for pollination with anything unknown, है ना। अब वो unknown होगा of course from the matching type only, ठीक। So when they have emasculated that flower, they will bag it, they will cover that plant, okay, this is called bagging, they will cover that flower, this is called bagging. After the stigma is matured, okay, what they will do, usually this is when both of them are maturing on the same time. So before the anther gets fully matured, they exactly emasculated it. Otherwise, so it will shed out, no? Okay? So up now, when the stigma will become mature, they will literally open that bag and fuse the stigma with the pollen of their choice, and then again they will bag it. Then again they will bag it, and then artificial hybridization is done. See? Another thing is with a flower which is for this cross pollination वाला. ये तो आपने देखा which was self pollination वाला. एक flower है which is like of only one sex, unisexual flower. So let's take a female flower, है ना? And female flower is only having the stigma. So what the scientists of this hybridization section they will do? Okay, they will keep this flower closed with bagging so that no outside pollen can come and fuse it. And when they are done with this pollination artificially, of course, then they will rebag that flower so that it can show the progeny of their choice. Okay, so this is some of the steps done in artificial hybridization, which are like bigger steps. Otherwise, तो भाई genetic recombination है, a lot of things is there, and we can go for recombinant DNA technology. ये तो बहुत ऐसी common सी thing है which was done from the time of Mendel. Now we are in the advanced section which we are not going to discuss here, don't worry. We will see in the biotechnology portion only if you want. So you can, since you can combine the genes even, if this one scenario is not possible easily, है ना? Big plants होते हैं which are not allowed in this bagging or all, so they can go for this other modes also. They can go for this other modes also, that is genetic recombination. So recombinant DNA technology is also one of this method. So today we have learned a lot of things and we are going for one more class of this chapter. Okay? So next class we will surely complete this chapter, and next class let's set the task what we are going to do. Okay? Next class we are going to first, so we have to complete this pollination because we have not done the agencies of pollination so far. Okay? So we will learn different types of pollination. Like pollination को भी broadly you can divide into two categories, abiotic and biotic. So always try to learn in a manner. किसी भी चीज को manner से learn कीजिए मेरे साथ, because this is not written in the NCERT biotic, but this way they ask you question, and at that time you will become blank. मैम ने तो बोला नहीं था, NCERT में तो था नहीं, है ना? So you can divide this into two categories, abiotic and biotic. Of course, abiotic are the agencies which are not having the life, है ना? तो wind pollination, water pollination, है ना? Wind pollination means anemophily and water pollination means hydrophily. They are the abiotic wala pollination. And when you come to the biotic sources, इसमें you are going to watch with this insect pollination, you are going to watch bird pollination, and you are going to watch bat pollination. Insect pollination को क्या बोलते हैं? What do you call to insect pollination?
और नेकोफिली
वो तो bird pollination है. What is insect pollination? Bat pollination को भी called chiropterophily, है ना? Yes, Shri, very good. Insect pollination को भी called entomophily. ठीक? So यहाँ तक be prepared from the NCERT. ये करके we are going for post fertilization development. और वहीं पे I will exactly explain you about the different type of methods how endosperm get mature. Like I will tell you about 3 methods: cellular, acellular, and the alluvial, and how this whole fertilized product is going to reach to the embryo stage and so far. Okay? And we will see how much we can do tomorrow, and yes, we will try to finish the chapter, and rest कोई भी fruit development या multiple fruit वाला even if it is left, so we will after this chapter अभी human reproduction, in place of doing that I am thinking to go for morphology of flowering plants. So after this chapter we will proceed towards morphology of flowering plants, because that is like अभी आपका mind frame is like in this type है ना, इसमें चल रहा है, तो it is best to go for morphology of flowering plants, and there we will be completing this. Okay? So tomorrow is going to be the last class for this chapter. Okay? So tomorrow means Monday. Okay? So take care everyone. Enjoy the weekend. See you on Monday. Okay? Bye bye. Take care.
Thank you, ma'am.